thing is, it keeps this job keeps you this position keeps you humble.
Yeah,
So
true.
we may or may not get another visitor today. May join us. I try to I forward the invite to him, or I forwarded the meeting to him. So he's I have a medical trainee, a medical resident who's working with me this month. So he may pop up on this.
Okay. I think Dr. Monica will not join us today, but let's wait for Dr. to get in and we speak.
Hello, Richard.
Oh,
Hello there, boss.
How are you?
I was just saying, I'm on our inpatient transplant service. We have like
A lot
21
of
in
work
at the
then.
moment.
Okay.
I had somebody get CRS this morning around 4 in the morning went to 105 Fahrenheit and atrial fibrillation, blood pressure's down we had to move in the ICU because the blood pressure was falling down to like the 60s. So,
hopefully he get better very soon, huh?
I'm counting on it.
Mm.
It was a haplo getting this is day 2 of so cytoxin's tomorrow.
Oh,
And
yeah.
they did not like the mismatch. So
okay.
there was a big T cell dose in the product. It was like almost 4 times 10 to the 8 per kilo of T cells.
Oh, quite a lot. Huh?
Yeah, big dose, and so they're reacting already. So we should be okay, hopefully.
Yeah.
So
good luck.
Yeah, I've been between just a lot of things happening in the United States right now, which are a little unusual. You probably may see some of the news, but they're completely shutting down or altering the government.
Oh, too bad, no?
I hope so. I think so. This is the new administration, and I was on I'm on an advisory board to the Health and Human Services, a national advisory board, and we've been postponed twice, and I wouldn't be surprised if we are discontinued from it. This is the new Health and Human Services, so they have a transplant advisory board to Medicare, and so a lot of what's happening in Washington D.C. is they're eliminating a lot of outside consultants. I don't think there's anybody in Health and Human Services have transplant experience, but which is why we're invited in the outside. But we'll see.
Yeah.
So
hopefully it's not too bad, no? It's not too bad.
Well, there's a lot of people running. I don't know if you see headlines for the United States, but they're froze a lot of NIH money. They've eliminated at the moment. They're eliminating a lot of supports to academic centers. So the NIH would fund research to an individual, and it would provide an additional 50% or 60% or 70% to academic centers to help support that individual. And across the country, they've announced that they're going to cut it down to 15%. And so across the U.S., many centers are unclear of how they're going to be able to support research going forth, but we will see. I know it's not a good time to have a political discussion, but I'm just saying that there's a lot of change happening in the United States right now. Justin was briefly on and off, but I see Leah's on, and Leah is probably an expert at the legal aspects of what's happening.
Hi, everybody.
Hello.
Well, I think we're just really trying to stay positive and trying to continue to move forward with what we can do. We are incredibly lucky in the United States. We have someone like you, Richard, who is a tremendous scientist and ambassador. And we're just going to have to keep moving forward with the amazing human beings that we do have and continuing to do the good work that we are doing. And we'll just have to see. Right now, I mean, a lot of this is stuck in the courts, and we suspect that it's going to be stuck in the courts for a long period of time. So I don't think that we're going to know exactly what will come of it for a while. I do think that you know, like part of a political strategy is just to throw a lot out there and kind of cause chaos so we can't focus on one thing. And I think we're trying just not to get caught up in that and just to stay focused on the work that we can do. Nothing has been cut as of yet because it's all stuck in the courts. So that's just what we're trying to do, is just stay laser focused on our work and the people that we're working with. And at least as of right now, nothing has changed.
Yeah, well, that's well spoken. So, you know, for the folks here, you know, in my new role, that was a big activity we talked about, because I'm now in the executive board of the American Society of Transplant Cell Therapy, and with the cuts, they wanted, should we make statements, not make statements, and we just said we should acknowledge it. I basically said we should acknowledge it. We're aware of it. We are going to pay attention, just as Leah, you just said. But there's other things going to happen too. Medicare, Medicaid. So we really got to wait until we, you know, you can't just jump at every word. You got to wait to see what. It may take a month or 2 or 2 months to really understand what the platform is, and then we really start addressing how we can, you know, address
And
this.
even if there is one, because right now every single thing that he has proposed for the most part is now being challenged in the courts. So it's really just, he's just trying to create a lot of chaos so that nobody can really focus on one thing. So we're just really trying not to get caught up in that. And until you know, the courts say any specific one way, we're just staying laser focused. But I feel really bad that we're spending any time on this, because I know that you have a lot of important presentations to get to. So
all right,
I'm
well, I was
going
just
to
saying, but he was asking what's happened. I'm just saying it's been an interesting 4 weeks or 6 weeks since we talked, because this is a lot of, as we're saying, a lot of discussion happening, and we all want to continue our good work and take care of all the people who need to be taken care of. And there's just there's a little in the U.S., across the U.S., there's just this uncertainty. So we'll see.
Yeah, well spoken. But in the meantime, we're in your good hands. So we'll let you take it away.
All right, everybody. So hi, everybody. We haven't chatted for a while, but it's like I said, it almost seems like, I know it was just a month ago we did have the follow-up from your team, but it seems a lot longer than 4 weeks. But either way, or so, what I was trying to do today is hopefully, does that show?
Yes.
All right. So I was going to talk about, you know, our 2 big meetings took place. In December was the hematology meetings, and a week and a half ago was the ASTCT, the transplant and cell therapy meetings. And so I thought I would talk again about, you know, give you some updates and snapshots from the ASH meeting as well as the tandem meeting. You know, this is reminding we have CAR Ts. This is by the way, just a picture I took, because this is the theoretical way in which you could do gene transfer and using nanoparticles similar to CAR T, but to treat heart attacks. Because there's now work coming out that when you have hypertensive cardiomyopathy that potentially puts stress on the heart, it will activate fibroblasts, and the fibroblasts will secrete molecules that can actually extend the heart. And so they've now developed these, basically the equivalent of a CAR T. So when someone has these, you know, stress, like type 2 myocardial infarctions or stress events, that you can actually potentially interfere with it with like a CAR T. So we're now going to get into protecting heart disease. So, and up here is just because, you know, for the whale, you know, I grew up in a whaling town. It was like famous for being one of the whaling capital of America. And this is called the Nantucket Sleigh Ride. When you get on board and when you do hook a whale, it's going to be one heck of a ride. That's how I look at what's happening in science, is we have so much going on. It's still going to be one heck of a ride. And I'm going to try and show you some of the new advances as fast as I can, but hopefully as clear as I can, that will be a benefit for your team. So reminding everybody, whoop, come on. Yeah, so they have The United States continues to have multiple advances in CAR T, and I know that's a big interest, at BDMS and as well as in Bangkok. Two well-characterized markers. We've talked about some of this before: CD19 in B cells, BCMA in myeloma cells. Here are some standard indications, that are currently approved. And these are the actual approved products that have been approved as of, November of this year. And these are from the major companies. The top four are for lymphoma. The two bottom ones are for myeloma. Well, this is to say that we're now got three more now FDA approved products. On the bottom is lifileucel, and it's a T cell product, a tumor infiltrating lymphocyte product. And I've talked at one of these sessions about TILs. But we now have obacaptogene autoleucel, or Obacel. This has got a very unique biology for adults with relapse refractory ALL, where they will generate a dose, and I'll explain in a minute, but it's got a fast off binding, which they think is going to cut down the risk of CRS, but it's also fractionated based on the number of the leukemia burden at the time of treatment. You'll either give one dose or two doses, one like early on and then one week later. That was approved in November of 2024. And then afami-cel is a T cell receptor transferred product for relapsed synovial cell sarcoma, where they have a T cell receptor that's virally introduced into the host T cells, and it is restricted on several HLA types using a MAGE, which is basically a melanoma-associated antigen, but it's also seen in other diseases. And so that's also been approved. And I'll tell you, we already have 4 patients, lined up to receive afami-cel. I showed you a slide before of what's happening in the world of cell therapy, which is these are a host of clinical trials. These are the approved products, and these are a variety of cell types that are being tested. And it's just showing the diversity, the extensive amount of work that's happening. Well, this is another one that I just found, which is, so these are all like, here's autologous CAR T, off the shelf donor allogeneic CAR T, allogeneic T cell receptor, or autologous T cell receptor. Like this is afami-cel right here. TILs, which is lifileucel. Natural killer cells are probably going to come soon, and a variety of others. Well, this is now showing what Big Pharma is doing. These are all the different potential clinical trials, and around the circumference are all the name of the major pharmaceuticals that are getting in. It's no longer just small biotech, but it is big biotech is buying up small companies or licensing technology. And so Big Pharma sees that cells are going to remain a market for the future. And these futures do include off-the-shelf allogeneic therapies, overcoming tumor microenvironment resistance, enhancing persistence of the cell product, increasing the ability to treat solid tumors, in vivo CAR, and some of these things I'm going to talk about hopefully in the next 50 minutes. How we can decrease toxicity and other cell types in constructs. And just mentioning constructs, there's a paper came out from Michelle Sadelain's group at Memorial Sloan Kettering. Carl June and Michelle Sadelain have both been granted the Lasker Prize for developing CAR T. But you can see our standard CAR T that we talk about, with the chimeric binding site from the antibody. Well, these are all variations of CARs and constructs that are under development right now in a variety of clinical trials. Just showing you the complexity with the goal that hopefully some of these are going to replace what we have and be either more efficacious or safer product overall. So stay tuned on that. And just reminding that, and again, you all know this CAR T continues to go up. This is an update from the IBMTR showing, oh yeah, we're going up. We're up by by 2022, we now have over close to 3,500 infusions. This is updates from the IBMTR. Well, if you look in the actual, information, this is on the web, a group that follows what's happening in pharmaceutical companies. As of last February, so we're now a year later, Kite alone has treated almost 20,000 patients. There's now over 40,000 patients treated with CAR T across Europe, across the United States, across China, and I know you're now doing this in Thailand. More and more countries are making this available. There's commercial indications. These are the standard, and then non-commercial indications are, potentially these are products that are research-based that are being now tested. Either way, and again, I think I've shown you our OHSU activity. This is our final update from the year 2024, where our number of transplants dropped some, went from 250 to 232. Our CAR T procedures and TIL procedures now went up to 91, up to 105, and our projections of where we are now after going into the first quarter is we're going to be closer to 120 to 130 for cell procedures as well as the 250 transplants. So right now we're averaging at OHSU a procedure a day, keeping us all busy. So what are we going to say? Before I go into some CAR T updates, I'm going to give a few transplant updates. And one of the biggest one that was hit was mantle cell lymphoma. And what the summary is that basically autologous transplant, which has been forever since like the 1990s, has been a mainstay in the management of mantle cell lymphoma. We're now showing that in the majority of patients with mantle cell, that upfront auto transplant will now be omitted. And it comes from the triangle data from Europe, as well as the ECOG data, which was updated in the late breaking abstract session. I'm not going to talk about the BTK. So the triangle study is someone you may remember hearing in the past. This was a plenary session at ASH about 3 years ago. Mantle cell patients on diagnosis were randomized to different arms. So they all got a standard induction with R-CHOP alternating with R-DHAP. But then the standard of care was to go to auto transplant and be observed. Well, they had two experimental arms. One was an auto transplant and then 2 years of ibrutinib maintenance, and then observation. And then a second experimental arm, a third arm, where it was they got ibrutinib as part of induction of the CHOP with CHOP, and then 2 years of maintenance and observation. I'd point out is all three arms still got standard rituximab, following as much as possible, following the Italian study that was published in New England Journal that showed that 3 years of every 2-month Rituxan improved outcome. And so what we now know is that what they showed back then is with that plenary session was with the early follow-up, it looks like that the worst arm was the patient who got auto transplant alone. And now with median follow-up of close to 5 years, this is auto transplant plus ibrutinib versus auto transplant alone. Statistically significant improvement in progression-free survival, failure-free survival. Same with ibrutinib alone without getting an auto transplant. And if you got ibrutinib, whether you got it with or without transplant, you did well. And if you look at overall survival, they're now showing what looks like a statistically significant improvement in overall survival if you get exposed to ibrutinib early in the management of mantle cell lymphoma, and that the absence of ibrutinib actually, was predicting a worse overall survival. So now the triangle data would suggest that we may not need to consider autologous transplant. The United States had another study that was sponsored by the lymphoma, national intergroup, And this was a late breaking abstract to the ASH meeting. And what it was is that there was no significant benefit to patients who got a transplant. And that potentially newer therapies were getting patients to remission, and that maybe that's what we need to do. And I'm going to show you what this was and what the schema was. Specifically, patients were newly diagnosed with mantle cell lymphoma. They would get their lymphoma would be sent for basically clone-seq to see if they have a molecular clonal abnormality of their immune globulin that could be followed sequentially over time. So did you have a clonal marker present or not? The majority of patients did. They then would be chosen to be, it was basically, you could choose how you want to induce, like you could use the regimen of alternating R-CHOP with R-DHAP. You could use R-bendamustine. You could use R-CHOP and then follow up with 2 cycles of DHAP. You could do different inductions, but then you would be randomized at time of transplant. But the randomization was based on whether or not you, to be eligible, the randomization was they, after the 6 cycles of induction therapy, they tested your blood and did you show evidence of the clonal rearrangement of the mantle cell lymphoma with a sensitivity of 1 in 1 million cells. If you were either indeterminate, or if you never had a clonal marker to begin with, maybe it was a necrotic specimen, maybe it just never showed up. They didn't want to deprive you of a transplant, so MRD positive, or MRD indeterminate, or after treatment, if you still had MRD positive, you got a transplant. That was RMC. But the key is if you were MRD negative, you were then randomized to standard care, which is transplant or rituximab for 3 years, or just rituximab, no transplant. So you treat them however you're going to treat them. If you have by adaptive technologies, if you could show a clonal rearrangement of the immune globulin receptor was available, that after induction, if you were MRD negative, you would be randomized. It turned out about 70%, or actually higher than 70% of the patients were ultimately MRD negative. So this is the interesting group. And it turns out, if you look at the difference for progression-free survival or overall survival, patients who were treated as a sign, like some people said, oh, I decided I want a transplant after all, or I decided I don't want a transplant after all. So this is a subset of those who were, this is intent to treat, and this was actually treated. There is no statistical difference between patients who got a transplant or didn't get a transplant. If you went by MIPI scores, like you could say, well, patients with very high, international prognostic index for the mantle cell, that's what a MIPI would be. Maybe those patients did better. Nope, didn't make a difference. If someone got R-CHOP alone with some couple cycles of DHAP at the end, or basically got a non-intensive like bendamustine rituximab, no difference either. So really, no major confining. The only place where they could see it is arm C. Is so if you had a if you didn't respond up front and you were still positive and you went to transplant, you could see that in the case that this is just for MRD positive patients, that compared to the MRD negative, you were going to have less survival and you're more likely to relapse. So they would still say, we don't know what the best therapy is for these patients, but they still could be considered for a transplant. But the big issue is, arm A and arm B is with or without, MRD persistence, and no difference. So now we have the original triangle trial showing us that ibrutinib up front means it would, takes away the need for transplant. And now we have the US study saying if you have deep responses, you don't need a transplant. So what does that tell us is right now is if we look at IBMTR, the number of transplants for mantle cell lymphoma by 2023 were already falling off, and they're projecting that maybe by 2024, maybe by 2025, it's just a concept that someone is suggesting. We may see that mantle cell for early auto transplant for mantle cell is now over. Going on, there was a lot of meetings about P53 and AML. There's 2 recent reviews talking about what do you do, because right now this is a big dilemma. We know that chemotherapy induction does not really effectively often benefit someone with mantle cell with AML. We also know that maybe we give venetoclax, but we're also finding a lot of those patients don't do well. And if you have P53 disease compared to other AML, they also do worse. And so this is a summary of these articles that we just published in January or December and January. But saying now that it isn't just to say P53 is bad. We now have subtypes of P53. Are you P53 deleted in one chromosome, or are you biallelic depleted? That also negatively impacts. And so this is a summary slide that came out that was sort of published, is if you have patient that you treat with P53 disease and you think you have disease control, and if you do an allo transplant, if they're have unstable disease within 3 months, they are going to do poorly. If they don't have GVH, chronic graft versus host, they're going to do poorly. If, on the other hand, you do a transplant with good disease control and they get chronic graft versus host, they're going to probably do okay. And in between, there's a whole host of small subsets saying that, like treatment-related AML, P53, molecular presence is cleared before the transplant. Does maintenance help? This is still, room for study. And what most people again say, we really do not know the answer. Some centers are just saying we're not going to transplant P53 AML anymore because I have responsibilities of maintaining good outcomes for my statistics, and because insurance companies in the United States, if you have really, really poor outcomes, they can just decide not to support your allowing transplant at your center anymore. So there are centers saying it just isn't worth it for me anymore, and they'll send them elsewhere. We do that's where I said it could impact your ability to maintain your qualifications of a center. There's new therapies. There's new targeted therapies for P53. People are trying to say maybe we can develop ways to which we can target the P53 before we go forth. We're just going to see activity. And also, can we maximize activation of the donor T cell product after the transplant for P53 disease? That may end up being a beneficial outcome for the patient. Another thing that was talked about at the ASH meeting is there was another Americans Blood and Marrow Transplant Clinical Trial Network, OHSU. I'm on the steering committee for that. There's 20 centers that represent the steering committee. 50% of the transplants in the country are performed by that group on the steering committee. But we have different protocols. And so the Stephanie Lee presented data from 1702, which was a study basically came out, which is, you guys in Thailand are very, very active using haploidentical transplant. You use haploidentical pretty early, as opposed to think about not pursuing unrelated transplant. And this was a question. That's why the study was asked is, when we do unrelated transplants, we can do a search. And the search will come back and say, you got a lot of molecular donors, and you're very likely to find a donor and go to transplant rapidly. Or you're very unlikely to find someone, or you're somewhere in between. So when we identify a patient, we send them for an unrelated search, we get this like over 60% likely to find a match, less than 20%, or basically 20 to 60 in between. And it turned out, so like, how hard should we look for unrelated transplants? So this is, again, the field's evolving. This started in 2017. There wasn't as much acceptance of post-transplant cytoxin. Well, it turned out that they ran the trial, and what the whole goal was to go to transplant early. Well, first of all, they found out that only a third of the patients really got to transplant within 6 to 12 weeks. Other centers kept pursuing, if you're in the likely, we're going to keep looking for an unrelated transplant rather than do mismatched, haplo. But ultimately, 50% went there. What they've shown is that over time, by the way, that people are now taking in the likely category, they're taking now 7 of 8s instead of full haplo. That's what we're seeing. But the fact is, like, this was the key. When you actually matched up the outcomes of haplo versus mismatch versus, fully matched, and whether you win early or late, there really was no significant difference. And so the message is, if you got if you want to do an unrelated transplant and you have a search that doesn't look promising, do not waste your time. Go right to an alternative donor, use a mismatched product, use PT-Cy. And finally, to mention is, I thought this was an important piece. So management of chronic graft versus host. So chronic graft versus host still is the thing that we worry about. Post-transplant Cy with tacrolimus and MMF has decreased the risk of chronic GVHD, but we still see it. And when you see it, steroids is still the #1 treatment. If you're steroid resistant, you have choices now. You have a choice of ruxolitinib, which is a JAK1/2 inhibitor. You have a choice of belamisadil, which is a nuclear pore, transduce transmitter or transfer center, a transfer pore inhibitor, and you also have axatilimab now, which just was approved also by the FDA in November of this year, November 2024. This is a monoclonal antibody against the CSF1 receptor. This is on colony stimulated factor 1. This is on monocytes and macrophages. There was in the Agave study. This was published. I think Dan Wolf, I believe, from Europe, was the first author. This was published in New England. Oh, here it is. Yeah, there he is. Dan Wolf is the first author. This was published in New England Journal. And what they showed is, interestingly, they divided patients getting the antibody into 3 doses. For whatever reason, normally we would think higher dose, better saturation, would have better outcomes. Well, it turned out the lowest dose was the most effective because it had the lowest toxicity. You give the antibody every 2 weeks, and there was a disease response benefit. So 75% of people with response. This is important. The median time to response was 2 months. So when you see someone with chronic GVHD and you start this therapy, it's going to be 50% of patients, they're going to respond after 2 months. You still got to keep the steroids going and not taper it too fast. But it's a good drug. It's a new drug for our collection for management of chronic GVHD. Like I said, it was approved in November. The company has not been able to manufacture enough yet to meet the demand. And so they held up release until they have their, biologic stores available. They will be available for release potentially next month is what I heard. Now, the other thing that was presented, this is what was published in this article that came out in the end of, I think this was synonymous, or this matched up with ASH. By the way, I'll also point out they're now developing an oral CSF1 receptor inhibitors, and they're now opening chronic GVHD studies. But this was the important aspect of axatilimab. When you look at the median time to response, like I said, overall is 2 months, but it can be up 6 weeks, would be 64%. But if you have gut-only disease, that actually did better than musculoskeletal with fasciitis. But skin was the lowest response rate. Skin we typically think is the higher response rate, but this may represent patients that went on study had already failed ruxolitinib or belamisotil, and they were very, very sclerodermatous. So potentially, if you have an inflammatory chronic GVHD patient, they may do well, but the sclerodermatous GVHD is probably only going to have about a 30% response, but it's still going to be a new drug available. Okay, 30 minutes down and 20 something minutes, 24 minutes to go. What about CAR T? Again, I always think, we talk about CAR T, and again, at ASH and at the tandem meetings, CAR T was a huge, source of activity. And I say it's a saga of success and failure. We see patients like this getting cleared within 30 days of lymphoma. We still are dealing with relapse. We still have the issue of proliferation, like the CD28 CARs rapidly expand, and the 41BB CARs seem to have better persistence. But we now have 5-year data emerging. Zuma 1 is published. Zuma 5 is now presented. And the Juliet data is about to go in and be published, hopefully in JCO, we'll see. Lots of discussion. There was a whole half-day session at ASH and a special session on relapse after CAR T. So, there's been several NIH sponsored symposium on what to do for relapse after allo transplant. This is the first that I've seen organized, completely organized to relapse after CAR T. They talked about intrinsic resistance, patients that will have, for whatever reason, biologically never respond. They talked about immune signatures, like what you can learn by looking at the immune profile of the T cell product before you infuse it. We looked at the expression of PD-L1 on the target, of the tumor target, or whether we turn off the T cells. Overexpression of, if you can overexpress tumor antigen, like high MYC is more likely to make you resistance. Of course, antigen loss is always, we're always aware of that. To be determined, there's going to be new mechanisms. I love this, is trogocytosis. And I don't know if you all on the phone or know about trogocytosis. There's now presentations coming out. This is really interesting. And it's based on how a CAR T cell binds its target. When the T cell receptor binds its target, you create what's called the immunologic synapse. The binding actually creates stronger adhesion molecules, and there's a tight junction activity. And by scanning electron microscopy, it's a ring structure of where the contact takes place, and it only requires 50 HLA peptide complexes per cell to lead to cell death of the target. What they now know in the immunologic synapse of the CAR T is a very convoluted membrane. The membrane builds up. The molecules are just almost haphazard. You almost need now, for the CAR T to kill its target, there's the target, there's the CAR T. You almost need 5,000 target molecules per cell. This is what work that's come out of, the work at Stanford from Crystal Mackle. And this is from Crystal Mackle's group is a publication. But what is trogocytosis? When you create this very fluid membrane, that's contacting, when the CAR T kills the target, you start to get the membrane starts to break up. The membrane actually of the target cell goes into the extracellular cytoplasm and actually fuses with your CAR T membrane. So now your CAR T cell is picking, like in the case of CD19 on the B cell lymphoma, you now have membrane that expresses CD19, the tumor cell dissolved, but the CAR T now absorbed membrane expressing CD19 antigen, and the CAR T, the neighboring CAR T, is killing the CAR T is killing the CAR T cell itself. So it's like you now have fratricide, but you did it because you absorbed CD19 from the target cell. That's what trogocytosis is. Lots of interesting biology. Does that change in what we do clinically? Not at all, but it's kind of fun to see the science. So what's coming? This is, again, we're going to talk about age. We're going to talk about diseases and expansions, as well as new science. I am not going to talk about progress in CRS or ICANN management. A lot of activity there too. And again, I showed this slide before, I think last October. This is data from ASH 2022 that's now coming out in publication. It's an IBMTR analysis showing when you take patients, getting either, this is lymphoma, getting either axicell or tisacell, age range 18 to 91, median age was 63. 4 age groups, and these are the 4 age groups. If you look at progression-free survival or overall survival, efficacy absolutely not impacted by age group. If you look at CRS, if you look at p-values, absolutely not impacted by age group. However, by ICANNs, the neurotoxicity, using the 18 to 54 as base, 55 to 64, maybe a slightly increased statistically likelihood of developing ICANS, but with 65 to 74 and over 75, marked increase in the rate of ICANS. So this is data that we've known, and this is from retrospective real world data. Basically, relatively anyone comes in. Well, now we have single center data from Stanford. So what they did is they presented the data from the octogenarian groups. So these are the patients over age 80. It's retrospective single institutional. And what they did is identify 21 patients that were over age 80. And they did a propensity match cohort. They found a 2-to-1 match of patients over age 65 that they could compare it to. What they showed is if someone was in that 80 group, you're more likely to get lysocell than axicell. You're more likely to get bridging therapy, and more people were avoiding cytoxin and fludarabine as a lymphodepleting therapy. They're using bendamustine. What they showed is overall response rate, 94% in the octogenarian, 92% in that, age 65 to 80 group. CR rates identical. Basically, no statistically significant difference in progression-free survival or overall survival. That's the same number. CRS was identical, relatively identical, maybe slightly increased, but not statistically significant. In their group, they did not see a statistically significant in all ICANS, but grade 3 ICANS definitely was higher in patients who were the octogenarian. The older patient needed longer time in the hospital, more likely to need the ICU, and more likely to die at 1 year of non-relapse mortality. But their conclusions, you should not rule out an octogenarian as a patient for CAR T therapy. Lymphoma is, this is reminding that lymphoma remains still the big target for us. High-grade lymphoma, this is where we were in the 90s. We used CHOP, and we know that R-CHOP improved overall survival. This is old data, and we now know that CAR T, if you fail R-CHOP, we now know that and you fail salvage and auto transplant, we know CAR T is good for you. Well, again, this is Zuma 1, 5-year survival after CAR T. We now know that, again, these are patients that were predicted to have no better than a 15% 2-year overall survival. That's 2 years at 15%. What we now know is you get close to a 50% survival at 2 years. And by 5 years, we're now seeing we're still seeing a 40 to 50% 5-year survival. And also not late relapse. Some of these patients could have relapsed and still be alive with further salvage, but overall survival is excellent. And progression-free survival, disease-free survival is excellent. They want to point out that again, we now know that if you relapse after R-CHOP and it's within 12 months, it is a standard of care to consider either axicabtagene or lisocabtagene as second-line therapy. Like I said, the choice of axicabtagene is a CD28 product, lisocabtagene is a 4-1BB product. A lot of people would prefer older patients for lisocabtagene, younger patients for Axicel. But what we do know is second-line therapy is standard of care for relapse refractory large cell lymphoma. What do we know now is very interesting is there's a new product called Firazyl or firacaptagene autoleucel. This is not FDA approved, but it's a CD22 product. This was a phase 1 institutional trial of patients who relapsed after CAR T or had CD19 negative large cell, thought never to be a benefit. And what they were looking for is the recommended phase 2 dose. They now have a dose of 1 times 10 to the 6 per kilo. With a median follow-up of 3 years, this is patients who failed CAR T or were not eligible for CAR T with a median prior line of therapy of 4. Overall response rate is 68% and 53% of complete remission. Overall survival of this really, we would say now, a very bad risk group. They failed CAR T. They weren't eligible for, other therapies. Overall survival at 3 years, almost 50%. Importantly, with this new product, this is CD22. We know CD22 has been associated at the NIH with high rates of ICANS. At least in this particular product, this particular CAR T product, no grade 3 or 4 CRS and no ICANS, no advanced ICANS also seen. So it looks like a pretty good product. There was higher non-relapse mortality, but that may be related to the patient population selected and not the CAR T itself. Another important study was a real-world assessment of Lysocell now in relapse refractory large cell, but also in patients with secondary CNS lymphoma. So this is going to be a select patient population. These are the ones that were initially restricted and weren't allowed for the original Axicel or Tisacel study. The original Lysocell allowed them to be treated. So this is now observational, real world, 36 patients with CNS disease, 30 were parenchymal, 6 were non-parenchymal, leptomeningeal. At the time of infusion, 97% had active CNS disease and 72% were felt refractory to the attempt to control the CNS disease. You can see prior intrathecal was 40%, prior radiation 50%. Impressively, overall response rate 64% with 53% CR rates. And if you like, the likelihood of responding in the 12-month duration of response was 55%. So this very high-risk patient population that we would have excluded originally are now felt to be a patient population of high unmet need and should be considered for CAR T therapy where appropriate. I can already look at my time and know I'm not going to get to myeloma or autoimmune disease, but it gives me a reason to talk to you guys again at another date. But let me tell you about what I think is really exciting data that's coming it's on research trial and we're about to be part of this research trial. So we know that, first of all, axicabtagene is now being tested in high-grade lymphoma up front. So MYC positive, double hit, double expressor. If you have this is a randomized trial. If you have somebody like that, you're getting treated with R-CHOP or R-EPOC. After 2 cycles, you get a PET scan. If they have PET persistence after 2 cycles, they're eligible. This is a trial that has reached accrual. The data has not come out yet, but we'll see the data probably next year at ASH. They're randomized to standard of care, which would be continue with R-CHOP and R-EPOC, looking to get 6 cycles or 8 cycles in and then see what you're going to do. Or CAR T after 2 cycles of induction. We don't know the answer, but we're moving, the field wants to move CAR T earlier. And then this is a company, Allogene. They've presented their data. I've shown their data for their advanced advanced lymphoma for off the shelf. But what they want to do is, what about using it up front? And this is a product called simacapagene insusegaladusel. And again, it's a CD19 CAR, standard CAR, but they knock out the endogenous T cell receptor, so you won't get adoptively transferred GVHD. And they also modify CD52. It's important because you can then use CD52 to help select your product later if you need it. Now, that's the first about the product. Now, the other thing is, David Kurtz was doing some work at Stanford. So we know about clone. I mentioned CloneSeq. This is the adaptive technology that can look for clonal rearrangements or point mutations in the VDGA region of a monoclonal antibody and of a monoclonal antibody rearrangement, and you can track your lymphoma. And so people have looked at CloneSeq as a sensitivity for large cell lymphoma at the end of R-CHOP, and they say, how many go on to develop relapse? And you can see one study was 25%, one was 17%. Well, David Kurtz developed a cell-free DNA assay for large cell lymphoma, looking at a variety of target genes that was originally listed at 40%. They've now refined that assay. They refined this to the point that there's a cell-free DNA that they think is 96% predictive of persistent large cell lymphoma. And they've now shown this was presented at the Lugano meetings a year and a half ago. They showed that, of the sensitivity, this is the probability of remaining free of disease from the start of treatment. If you were negative for cell-free DNA, you're pretty well assured that you're not going to relapse. If, on the other hand, you were positive, you could see at 1 year, 70% of patients relapse, and by 3 years, it's close to 90%. And so they feel that this is a very active assay, and they've now basically licensed that assay out for assessment. And so this particular company, Allogene, is running the Alpha-3 trial. So we're going to be part of this trial. And what it will be is patients will get referred to us when they're on R-CHOP or when they're getting R-EPOCH. Typically, probably by third or fourth cycle. We'll tell them about the study. If they say they want to be screened and be part of the study, we'll enroll them. After 6 cycles of R-CHOP or after R-EPOCH, they will then get a blood draw. And the blood draw is basically, the blood draw is going to basically ask the question, are you positive or not? If you are positive, if you're not, you've got that, you could be a good chance that you're going to be cured already and you're left alone. If you're positive by that assay, by the phase edge sequencing assay, you will be randomized to immediate CAR T using an off-the-shelf product or observation. If you're randomized observation and you have late relapse, you're sort of guaranteed a crossover. So you will get a CAR T later, but this is asking, can we consolidate somebody after CAR T? So I'm going to skip that for the matter of time. I'm going to say high-grade lymphoma. There's all these type of lymphomas. When we say large cell lymphoma, there's many different type. What we're now showing is, and there's some IBMTR data, that if this group of here, this particular group, and maybe this one here, and maybe these 3, all are more likely to relapse than this is after CAR T. As I said, we're learning a lot about CAR T. We're identifying even subtypes of large cell lymphoma that's more likely to do poorly, and it gives us something that we're going to think about treating. Just to really very fast mention follicular lymphoma, Zuma 5. As I said, we now have Zuma 1, which is large cell 5-year data. We now have 5-year follow-up data from AXA-cell for relapse refractory follicular lymphoma. And you could see that the overall response is excellent. You know, in these patients, all patients in follicular lymphoma or marginal zone lymphoma, a little bit less, but almost a 95% response rate in follicular lymphoma getting CAR T. Median follow-up now is over 5 years. And what it looks like is this. Overall survival versus this is the marginal zone versus follicular, and orange is all patients. Overall survival at 70% at 5 years. Lymphoma specific survival, basically living without relapse, at over 80%. And yeah, so yeah, the rate of lymphoma specific death now is only down to 16% if we look at the type of death. So honestly, that was considered dying of a complication of CAR T. What was felt like maybe you were so immune suppressed you got COVID, maybe you had CRS or ICANS, or you died of relapsed lymphoma. The fact is, these are like superb data, and we would think that follicular lymphoma, everyone is trying to jump to bispecifics, doing what you can, but CAR T for large cell lymphoma is, in follicular lymphoma and marginal zone lymphoma, is excellent. So I'm going to stop there. That's slide 47. I had 20 more slides talking about AML and myeloma, but maybe that gives us a reason to talk again. So I'm going to stop sharing. And I know I talked a little longer than I meant to, but sorry about that. Questions for you? Any comments, questions that I could share before we adjourn and next month meet again? But this is ASHN ASTCT update. A lot is happening. I still love this field because it keeps happening. It keeps changing. Dr. Surapo.
Thank you very much, Richard, for your very comprehensive and very inspired talk. Yeah.
Well, what I will do, as always, I will send Jenny the slides, a copy of the slides. So even if we don't get to talk about them on your own, you'll get a chance to look at a couple of the slides that was updating ASH for myeloma, updating ASH for lymphoma, and then a little bit of autoimmune in the future.
Yeah.
Thank you.
Very good. Very good. Thank you.
Thumbs up, clapping, hearts. It makes me feel good. Happy Valentine's Day to everybody. And here's Dr. Denny. Yeah, well, joining Leah.
Sorry I came a bit late, but I got the end of that. That was terrific. Thank you, Richard, so much. I hope you're holding up well with all the changes.
Yeah, we talked about that a little bit, like the first 5 minutes about Leah and I, and Leah did a wonderful job of just saying, you know, there's a lot of like scare stuff going on, but we just have to be patient and keep our eye on the prize and keep going forward.
We will persevere.
And so I'm looking forward to continuing. I will send back notes between just a lot of stuff going on and going to the meeting and doing some other stuff. I didn't get back to send you my final thoughts on the work plan. I like the work plan for the year. We will work on, I will independently send notes about the group discussions, like when we talk about the all BDMS lectures, we'll come up, maybe get an all myeloma lecture and all, like covering everything, so to make it more interesting. And we'll also start planning, I think, one more trip, I guess, which I look forward to in the next visit.
Okay.
Thank you.
And whatever I
anything
do for you, I look forward to continuing.
If this leads to any questions or ideas from Thailand, please let us know. I think Richard spoke for the time, but if there's anything that comes out of these ideas and updates, let us know.
Yeah. Yeah. Thank you very much. Thank you very much. Yeah.
All right.
Thank you all. Thank you, Richard. Good to see you.
Thank you for your time, Richard.
Well, thank you for listening. I hope I hope this is all useful when I do it. As again, I actually get a lot of benefit out of this. This helped, just what you say. This helps me because it made me go back and look at the action, the tandem meeting, and update what the really important stuff, because this is going to change our practices. This is going to make us better for the future for our patients. So that's what we try to do.
Thanks
Very
all.
good.
Thank you.
Yeah, we
Thank
learned a
you.
lot.
Goodbye.
Thank you, Richard.
Thank you.
All right, everybody.
Bye bye.